a little bit about other options. I think we're waiting for a lot of the additional immunotherapy data, such as the durva-tremi arm of the Caspian trial to read out. But I think we have two options. First line, we discussed second line options. And we really want to just kind of look at the literature and see what's next. And I'll pick on Jamie because she was involved in this trial, not directly, but she treated some of the patients on the DLL3 targeting therapy with Rova-T. And you want to talk about your experience with this and I think why it's so difficult to develop drugs in small cell?
So DLL3 was a hugely exciting target and still is. There are multiple new drugs in development targeting DLL3. As a part of the Notch pathway, developmentally makes sense to target. And I think we saw both an expression-based response, so immunohistochemistry for DLL3 enriched for response to the agent Rova-T, though we were just limited by toxicity. The drug, while effective, was exceptionally hard on patients, including severe life-threatening toxicities, notably edema and effusions. So to my understanding, the development of the initial DLL3 ADC is done, but I think we're still enthusiastic about DLL3 as a target. It's expressed particularly in certain subsets of small cell and up to 2/3, and this should be a promising target for the future with other immune-directed approaches, bispecific antibodies, CAR T cells, things like that.
And Tawfiq, I know you've been involved, in the lurbinectedin experience. I mean, when I look at the data, it's pretty impressive, and you've been very involved in these trials. I'd love to hear more about this agent and where you see its future.
Yeah, so, well, just in the interest of full disclosure, I've actually not been directly involved with the trial, but I've been following the data, and some of the investigators there, like the rest of us, were good friends. So I think the lurbinectedin is a very interesting compound, at least based on what we now know from published, presented data at meetings. This is an agent that seems to be particularly effective in patients with chemo-sensitive disease. And the mechanism of action, I think, also is rational for small cell, which we now know to be a transcriptionally addicted tumor. So this is an agent that works by inhibiting RNA polymerase II function and therefore preventing transcription to occur. So the earliest evidence that this could be an agent that we use in our patient population came from the doublet trial looking at combination of lurbinectedin with doxorubicin. And that was a carryover from the attempt originally to develop this in sarcoma patients where doxorubicin was the backbone. So they just layered this on. And they saw some signal there, and then that led to the launching of the Atlantis trial, which is the randomized phase 3 trial of that combination versus standard of care. In retrospect, the observation was then made that perhaps for this group of patients, maybe you don't need the doublet. So they went back to now do the single agent trial, which is now what is looking really promising and was not that different than what they saw with the original single-arm phase 2 trial with the doublet chemotherapy. With this, the overall response was about 35%, but when you look at patients with chemo-sensitive disease, that's actually close to 50%. For those with chemo-resistant disease, it's low double digits, maybe around 20%. But regardless of whether you're chemo-sensitive or refractory or chemo-resistant, there is something to be said for this agent in relapse small cell. The disappointment that I see with this is despite the high response rate, PFS was still around 5 months. Overall survival around 9 months. I think for the chemo-sensitive, perhaps up to about 12 months, which is still better than what we have now. Whether this will become standard of care that we can use for our patient outside of the ongoing clinical trials, I think remains to be seen. We'll know maybe later this year or early next year whether or not there will be regulatory approval for it or not. But that is where we stand with this agent.